in order to make sure that we're all on the same page, my understanding is that we might delve a little bit into the biology of ethics, research ethics, and specifically genetic editing. So there have been a lot of in the news cases recently about fetuses being genetically engineered by a Chinese scientist, and so that's what I'm going to tell you about now for the next 5 to 10 minutes. Basic background if you're not already up to biology. So I want to talk about a natural form of genetic editing, which we call mutation. So if you look around the room, you'll see a lot of genetic variation among humans. Right? Hair color, eye color, height, so forth. And each of us, if we took any random pair of us, are something like 99.9% genetically identical. If you zoom in on our chromosomes and you look at the structure of and the sequence of the nucleotides, the letters that make up our genomes. So we have 23 pairs of chromosomes each. We have billions of letters of DNA that spell out the codes that encode proteins that make us what we are and help us be alive and do all the things we do, like study philosophy, pipette things in research laboratories, breathe, eat, digest, reproduce, and so forth. So here is an example of a double helix, this physical structure of DNA, and I've just listed a few letters. DNA is made up of 4 nucleotides or base pairs, A's, T's, G's, and C's. So all of the code for life is encoded by a long series of A's, T's, G's, and C's in our DNA, and somehow the book of life is possible using just 4 letters, not the 26 we have in English, for example. So that could be one individual, and another individual, another one of us in the room, might have generally the same DNA sequence, but there's one difference. Right? In that 4th position, a C in one individual might be a G in somebody else. And it's those rare differences, again, 99.9% identical, the same in DNA sequence, any 2 of us at random. So there are, between all of us, differences in our genes. So in other words, we're all mutants. Because all of us are genetically different. And it's impossible to say, usually, at least in this scenario, which individual is the mutant. We would have to know, was G originally there and it became a C and that was the mutation, or was it the other way? So here I've drawn it that C was the original version of this genotype or this sequence of DNA and that G is different. But in practice, generally, we don't know which came first. So mutations happen. They happen all the time. We're all products of mutation. In terms of genetic editing, some mutations are common, some are rare, and it turns out that ones that we anthropomorphically think are useful are really rare. So here's a great example of a mutation that benefited us. On the left is the ancestor to modern corn, which is especially useful to talk about in the Central Valley. Way back before corn was domesticated, a couple of millennia ago in central Mexico, teosinte was the plant. And it has this really tough seed coat around the kernel of corn that humans couldn't actually physically access. And you could break it apart, but you smash the kernel inside. And so it was not a good food stuff until one day, by chance, one mutation happened in one gene, the TGA1 gene. You don't need to know what that means. It really means teosinte gloom architecture, but I'm not a botanist, so I don't know what a gloom is. I'm a geneticist. There's one nucleotide difference. There's one mutation. In teosinte, there's a G letter at one specific spot on their chromosome, and in maize, there's a C. One mutation allows the seed coat to open in the plant that was then became maize, domesticated through breeding in central Mexico, and became a hugely important foodstuff for everybody, at least on the West Coast. Okay. One nucleotide change, and it happened in nature. A mutation, the plant was growing, DNA sequence randomly changed. We can talk later if you're interested about how that happens randomly. We're not all perfect. And so now we have a useful to humans mutation on the right, just one mutation. So one of the things that genetic engineers like to advertise about the benefits of genetic engineering is that it's faster, because this process took who knows how long to find that random plant that had this one very subtle difference that happened to be useful to humans. So, an enterprising company in Washington State, the apple capital of the United States, if not the apple capital of the world, genetically engineered an apple to not brown when you cut it. This is fantastic. McDonald's loves this because then they can package the apple slices in the baggies and they don't brown as quickly. They don't have to throw out as many apples. Right? So it's pleasing to the customer. So how did they do this? They genetically engineered the apple to remove the enzyme, one protein encoded by one gene. They disabled that protein, and it's a protein that causes browning. So it's a mutation. Scientists introduced the mutation to the apple. It didn't happen naturally. But this sort of mutation that causes apples not to brown anymore could happen naturally. It would just take a really long time for apple growers to happen to slice all of their apples, millions and billions of apples, and find the one that doesn't brown when you cut it. Then they would be able to produce exactly the same product. We just did it faster because it was done in a laboratory by scientists with a directed mutation. So the point is there's a difference here, or is there? Is it important to wait until a random mutation happens? Is that okay? Versus scientists using directed mutations. So that's a thought I will leave you with. Which of these is a genetically modified organism? Both. That's the question. Right, is it one or is it the other or is it both? So we could talk a bit particularly about the definition of what a genetically modified organism is. The last thing that's really important to point out, I think, for background for what we may wind up talking about tonight, is whether or not a mutation could be passed from parent to child. So there are two different parts in the body, the soma and the germline. The soma is the material that makes up our physical bodies. The germline specifically is where our germ cells, our sperm or our eggs are. So the only mutations that you can pass from you to your kids are the ones that are in either your sperm or your oocytes, your eggs. That's the only material from your body that passes into the next generation. So, and a good example of this, just to make this a little bit clearer, is if you get a skin cancer. I'm pretty clearly Caucasian and Scandinavian. I'm very prone to skin cancer, so I worry about these sorts of things all the time. Ultraviolet light hits one of my skin cells, causes a mutation. That might cause me to get a tumor growing on my skin. Those are not heritable, because my skin does not pass to my kids. But if a mutation happens in the sperm or the eggs, then that can be passed on. So there's an important difference, maybe, depending on what we talk about later, between mutations that happen in somatic tissue, the body, versus mutations that are introduced into individuals that are also heritable. That introduces the question of whether or not there's the risk of spread of genetic engineered DNA through a population or through society, rather than just being in the individual that that mutation occurs in. There are potentially some beneficial uses of genetic engineering. Here's just one quick example. Sickle cell disease is caused by a single mutation, an A to G mutation there in red, in the beta globin gene. Causes the red blood cells to change shape. They cause lots of problems, especially in the African community. And so there's currently a clinical trial underway because you could introduce a genetic engineering approach, I suppose, to target the tissues in the human that develop red blood cells. And we could talk about whether or not that's a good thing or a bad thing. Is that an ethical approach or is it not? Finally, there's a lot of interest and perhaps fear in society about what we can and can't do with genetic engineering. And so maybe it harkens back to Hitler and Nazism and trying to breed a super race or select only individuals to breed that have the ideal characteristics. The problem with doing this, one of the problems with doing this, is that most of the traits that people might care about, hair color, skin color, eye color, height, weight, IQ, are not just caused or influenced by a single gene. So there's no way to remove one gene or disable one gene in an individual with a mutation and have a huge effect on height, weight, disease susceptibility, in many cases, IQ, and so forth. So it's really difficult to predict the effect of any mutation that we might introduce, and that is definitely a concern. So mutations happen to all of us all the time. Some of them are good, some of them are bad. They only get inherited from parent to child if they're in the germline, so not all mutations can be inherited. And many of the traits that we might be interested in changing in humans or in any other species are usually controlled by multiple genes, multiple parts of our DNA and our chromosomes, so it's kind of hard, de novo, without more information to impact those traits by mutation. Finally, a plug for something else, I don't know if I'm allowed to do this, is that there's also the Institute for Media and Public Trust here at Fresno State. I'm not involved with this, I just know about it, is putting on a workshop or a panel and a keynote on how science is reported in the media. So if you're interested in that, there's more information that you can find out about online at the Media and Public Trust website. And with that, I'll pass off the presentation to our next individual. And by the way, I recorded the slides in my voice now, so if you want to share this or listen to it again, this presentation, this part of the presentation will be on YouTube if you need to refer back to it.